We're the Unit Processes Analysis Coagulant Subteam. I'm Sarah.
And I'm Ada.
So our goal for this semester was to determine a minimum coagulant dosage and observe the effect of high coagulant dosage on turbidity. Coagulant can be a significant operating cost for agua clara water treatment plants. Establishing a minimum coagulant dosage for a desired effluent turbidity minimizes the operating cost associated with the purchase of coagulant. By observing the effect of high coagulant dosage on floc formation, we will better understand the interactions between coagulant and primary particles. This knowledge could potentially be applied in future experiments involving the flocculation and sedimentation processes. Coagulant is added to dirty water in the water treatment process because it causes particles of material such as clay or bacteria to stick together, resulting in larger, heavier particles which will ultimately separate from the water during the sedimentation process via gravity. Flocculation agitates the particles so that the number of collisions between particles is higher, resulting in more successful collisions and more particles stuck together. Coagulant and flocculation are very important to the sedimentation process, and it is these factors that we hope to gain an understanding of through our research.
So after the flocs are formed they enter the sedimentation tank, where they settle to the bottom of the tank and form a floc blanket. So a floc blanket is a fluidized bed of solids, and it is important for the blankets to be stable and uniform in order to efficiently remove as much particles from the water before it enters the filtration system. So in the lab, we are using a tube settler, which is a model of a full-size sedimentation tank. And the tube settler has a floc hopper to help maintain a constant floc blanket height, and a plate settler to give the flocs more time to settle. So for our lab, we are using this experimental setup, and we have a total of 4 pumps. So the first pump is a water pump that pumps clean water into the system, and then there's a clay and coagulant pump that allows a constant concentration of clay and coagulant to enter the system, and lastly the waste pump pumps out the water after the effluent turbidity is measured. So for the other UPA subteams, they are using 4.5 milligrams per liter of coagulant, while we are varying ours from 0.14 to 64. And so we are testing a total of 10 coagulant dosages using a geometric progression, which means that we are increasing our coagulant dosage by a factor of 2 each time. So previously, a grad student named William Pennock conducted a very similar research where he varied his coagulant dosage from 0.05 to 98. And as you can see in this graph, as the coagulant dosage increases, the effluent turbidity decreases, which means that there is an improvement in the performance of the floc blanket. However, as it reaches a certain point, the effluent turbidity increases again, which means that the floc blanket is failing.
So Monroe has told us that he believes that the coagulant is more inclined to stick to itself than to the particles in the water. This means that if the particles are more than half covered in coagulant, it is more likely that the coagulant will stick to itself and simply transfer from particle to particle, instead of causing particles to stick together. Such a phenomenon would decrease the percentage of successful collisions, and floc formation would be impaired. If this is the case, we would expect to see a significant increase in turbidity after the critical coagulant dosage.
So for the majority of the semester, we mainly focus on setting up the experimental apparatus, so we're only able to test 4 different coagulant dosages. And as you can see in the graph, there's a lot of fluctuation in the data, and there's a lot of times where the effluent turbidity is 1,100, which means that the turbidity is too high for the turbidimeter to read. So that's why the team decided on focusing on where the turbidity is at steady state. So while we were running our experiments, we noticed that there's a lot of gelling, which means that there's a lot of places in the tube settler where there's really dense floc blankets. And we believe that the source of this problem was that the influent turbidity was around 700 NTUs for about 30 to 40 minutes in the beginning of the experiment. So the team solved this problem by modifying our experimental apparatus to allow the influent to directly go to the waistline before it reaches the target of 100 NTU. But even after that, we noticed that the floc blankets are actually going up the place settler rather than into the floc weir, which still causes the floc blanket to be unstable and not uniformed.
So here I have a chart summarizing the effluent turbidities at the various coagulant dosages that we have at the steady states. And you can see that effluent turbidity decreases as coagulant dosage increases, which is what we expect, and that corresponds to the Williams-Pennock graph also. However, you'll notice that the effluent turbidities are far below our 3 NTU target, and we're not totally sure why. And we were unable to establish a minimum dosage or observe the effects of high dosage from the data because the coagulant dosages that we measured, the turbidities at were not low or high enough to determine this. So, our future tasks include running trials at much lower and much higher coagulant concentrations, evaluating our experiment setup and analysis to determine why the effluent turbidity was so much lower than expected, determining the cause of gelling in the sedimentation tube, and modifying our PID values to improve stabilization of the influent turbidity.
So if you have any questions or recommendations, please feel free to email us. Thank you.